All right, good morning, everyone. My name is Lena Balonen, and I'm the Executive Director of the Global Soil Biodiversity Initiative. And this is the organization that runs these GSBI Speaks webinar series. Today's webinar will be on soil inoculation. It should be a very fruitful discussion. Before we start, I have a few housekeeping items. First of all, this meeting will be recorded. If you're not comfortable with that, we'll just have to ask you to sign off, unfortunately. So all these webinars will be posted to YouTube. Then people that are unable to attend the webinar, mostly due to time zones, will be able to watch them, as well as educators and others that may be interested. The second item is that all questions will be run through the Q&A. So you'll see the Q&A box at the bottom of your Zoom, and that's where you can ask any questions. So our moderator, Martin Hartmann, will be monitoring the Q&A, and only questions asked by Martin out loud will be answered. So we will not be answering any questions typed just out loud. So with that, I'll be turning over to Martin, who will be our moderator today. Thank you, Martin, and thank you to our panelists.
Thank you, Lena, and also a very warm welcome from my side. Happy to see so many people joining this webinar on soil inoculation, which I think is a very exciting and at the same time very controversial topic. So I'm looking very much forward to hear the input of our panelists and then also have a great discussion on this. I will first do a very short introduction and then introduce our panel before we then move over. So I will quickly share my screen. And I decided to start the introduction basically with this slide as an image to show that not so long ago, this is how we basically depicted, the plant growing in the soil and the soil largely being a black box with some nutrients in there that the plants require. But by now we know that the soil is full of life, most of it actually microscopic. So for example, 1 teaspoon of soil has around 10 to the 10 million cells in it. And these microbes are, or soil life in general, is incredibly important. I just want to highlight a few functions that are important for the soil plant system. Maybe starting off with the nutrient cycling here shown as a nitrogen cycle, going from nitrogen fixation to the conversion to ammonium and nitrate and back to the nitrogen. Very important, globally important also because, for example, nitrogen fixation, we know there is an estimate around 100 million tons of nitrogen fixed through biological fixation globally, which is pretty much the same amount as we have mineral fertilization globally applied. Beyond nitrogen cycling, they are very important in mediating stress tolerances of plants, for example, mycorrhizae by helping acquiring water and phosphate, or also different kinds of bacteria that produce plant hormones to stimulate plant growth and basically foster stress tolerance to abiotic stresses. But also in terms of biotic stresses, we know they are important regulators of pests and diseases, for example, different kinds of fungi or bacteria that regulate insect pests or other pests that we have in the soil system. Another important function is these organisms are enzymatic powerhouses, basically to degrade all kinds of pollutants ranging from pharmaceuticals to pesticides or even plastics that accumulate in soil. For example, here shown Aspergillus and Rhodococcus as 2 very prominent examples of this. And then last but not least, they are very key for climate regulation, visualized here by the methane cycle, for example, methanotrophic bacteria that consume methane, or on the other side, methanogenic archaea that emit or that produce methane. And this also at a globally relevant scale. For example, we know that microbial activity is responsible for around 75% of the methane emission, but at the same time, they also consume around 60% of the methane. So I think there's no doubt, and we all agree that soil life is important for the functioning of a plant soil system. And we are very interested in how can we promote and bring these important functions into place. And this is where the whole concept of how do we manage soil biodiversity, how do we promote specific organisms comes into play. And I would like to talk about very briefly 3 different concepts of how we can bring the right organisms to the soil, to the rhizosphere, or maybe to above ground plants. And the first concept is basically by managing the system, managing to basically modify the environment, changing the habitat conditions, for example, through agriculture or forest management, and thereby enrich for microbes that we want. These are very strong drivers, but at the same time, it's very difficult to steer these communities in certain directions. So we lack the understanding of this complex interplay between mentioned practices and soil biodiversity. The second concept is the host. So introducing a host that is associating with particular microbes or other organisms that we want to have in place. This is a relatively new field and it needs a lot more research to see how we can direct this and how effective this mechanism is to put the organisms in place. And maybe the one that's the oldest and maybe also most directed approach is really soil inoculation. So to inoculate with organisms, mostly microorganisms, either individually or maybe in simple consortia, how to bring it in. And that's on the one hand, commercially, something very promising. And as I said, it's very directed, but at the same time, it comes with questions. Do these organisms that we actually introduce into a system, do they establish? Do they perform the function we think they do? And are there any risks associated with this? And this is exactly a kind of the range of questions that will be addressed by our panel today. Which is, I'm very excited actually to have these four excellent speakers with us today that will introduce various parts of this topic. We will start off with Colin Averill. He's the founder and CEO of Funga Public Benefit Corporation in Austin, Texas. And there, Colin and his team, they translate science into action to restore below ground fungal biodiversity as a natural climate solution. And today, Colin will talk a bit about his rather long history of academic work he has done related to soil inoculation, and then provide insights how they try at Funga to bring these approaches to scale. After Colin, we will have Adriana Corrales. She's a lead field research scientist and the director of the Underground Explorers Program at the Society for the Protection of Underground Networks, better known as SPUN. And this is a non-profit organization focused on the conservation of mycorrhizal fungi. And Adriana will talk about live soil mycorrhizal inoculation experiments for restoring and conserving tropical tree species. So the first two talks will mostly be about success stories, how we can successfully use soil inoculation. And this will then be contrasted a little bit by Liz Koziol. She's an assistant research professor at the University of Kansas and the owner of Microbloom. And Liz and her team, they specialize in developing ecosystem-specific inoculants of AMF, or vascular mycorrhizal fungi, for agriculture and restoration. And indeed, Liz will talk a bit about the downside, maybe the widespread failure actually of commercial inoculants, and why it's very important that we improve the quality control to make these products successful. And last but not least, we will have Miranda Hart, professor at the University of British Columbia in Okanagan. Miranda and her group investigate the use of biofertilizers from best practices and a conservation lens. And interesting, Miranda will talk about ecosystem risks associated with actually introducing species from the outside into a system. So with these four speakers, we will basically have the whole range covered and hopefully lead to a very engaged and interesting discussion. So we will hear first of around 5 minutes from each of the speakers, their view on the topic, and then we will open the Q&A round. And so please don't hesitate to type in your most pressing questions into the chat. So with this, I'll stop sharing my screen. I would ask Colin to turn on the camera and share his screen. And it's great to have you here, and the floor is all yours, Colin.
All right, thank you so much for the opportunity to be here. So my team studies the forest microbiome. And the major research question we've been chasing for 6 years is, what does a healthy forest microbiome look like? And I realize the word health is squishy, but it's also useful. I hope it becomes clear why soon. So in some ways, we're closer to answering this question for people than we are plants. So the Human Microbiome Project has really led here. Your body is a microbial ecosystem. Each of us houses an incredibly biodiverse community of bacteria in our gut, and this has a profound effect on our health. And this was first discovered by medical microbiologists. They use DNA sequencing to understand which bacteria inhabit the guts of thousands of people. And importantly, they also have paired information on those people's health and disease status. So are they sick, and if so, with what? What's their blood pressure, digestive health, mental health? And if you combine all of that information, you can identify combinations of bacteria linked to health and disease states. And so these analyses became a roadmap for microbiome transplant therapies, some of which now represent standard of care. And so my team asked, what would it look like to take this Human Microbiome Project approach, but apply it to the forest? Could we identify fungi linked to tree growth, to restoration outcomes, to carbon capture? And so over the past 6 years, that's what we did. And we had 2 major findings. And so first, by DNA sequencing over 300 European forest monitoring sites, we found which fungi inhabit a forest is linked to 3-fold variation in forest productivity and carbon sequestration. This work is in some ways analogous to the human microbiome work, correlationally linking microbiome information with health and disease outcomes. Again, it's correlational, but if the underlying mechanisms are causal, these findings suggest we may have a new approach to identify the right fungal communities to drive outcomes for agriculture, forestry, ecosystem restoration. And that's important because while there is absolutely strong evidence that soil inoculation can work, the responses in academic trials are wildly variable. In 2022, we published a meta-analysis of 80 experiments where people did some sort of wild microbiome treatment like soil inoculation. We saw an average biomass response of plants of about 64% across studies. That's a lot, but there's also huge variation here. There are plenty of zeros. There are also plenty of big effects. Others have studied commercial fungal inoculants in restoration and agriculture. The average effect size found in those studies is about 0 by and large. Existing commercial inoculants generally don't work. And so there seems to be something here, and this is not really new. There are reports of successful soil transplant work going back almost a century, and pioneers like Dr. Koziol, Dr. Corrales on this panel are putting this approach to work in a restoration context. There's something about wild microbiology existing commercial inoculants fail to replicate. But the question remains, how do you identify the right donor ecosystem and with the right microbial community for the place you're trying to restore? I think the next big step forward here is actually putting the correlational approach together with the manipulative inoculation trials, explicitly leaning to wild, biodiverse, and complex soil communities. But that's very difficult. To do that well, you need to run dozens of field trials, survey hundreds or thousands of ecosystems, and have a long-term focus. I quickly discovered that was beyond what I could accomplish in academia. And so almost 3 years ago, I launched Funga. And so Funga is a startup and public benefit corporation. It exists to do the science to bring soil microbiome restoration to scale as a natural climate solution. So we reintroduce fungi to forests to accelerate tree growth and carbon drawdown. We've chosen to bring this to scale first in loblolly pine, which is one of North America's largest timber markets. We chose it because this pine is native to this region, so there's native fungal biodiversity to work with, because timber plays an important role in decarbonizing the built environment, and because we already know timber clearcuts degrade soil fungal biodiversity. This is what a 500-acre clearcut looks like in southern pine. We know clearcuts degrade soil fungal biodiversity, and we've confirmed this happens in southern pine. Once they cut at this scale, they then often burn these sites to clear debris. They'll apply herbicide from a helicopter to clear out remaining vegetation, including naturally regenerating pines. And finally, they'll often plow it, which is a massive soil disturbance. And so when you see this, it's easy to imagine how some soil biodiversity is lost here and how active microbial restoration can drive a real outcome in ecosystems undergoing this level of disturbance. And so at Funga, we've now merged this correlational and experimental sides of this research to identify our best estimates of what intact biodiverse communities of fungi look like in this system, that also drive outcomes for forest growth and carbon removal. And so this is the geographic range of our focal tree. We've collected and sequenced over 1,000 soil from over 1,000 forests to build our correlational map to identify which fungi are linked to growth and carbon removal and also represent intact and biodiverse communities. We've then tested a subset of candidates selected by our data platform in the greenhouse, and then we test them again in the field. About 85% of inoculants we've advanced to field trial have driven a positive outcome for either tree growth or tree survival. And I want to emphasize all of this is an iterative process. Projects and field trials allow us to hone and refine this approach. And furthermore, it's not one size fits all. We work with wild and complex communities. We don't put the same organisms everywhere. It's prescriptive based on geography and environment. And so to close out, I want to, this is to illustrate that it's iterative. And to close out, I want to return to this question. So what does a healthy forest microbiome look like? Or a prairie, or a mangrove? I love this question because it generalizes across ecosystems and functions. In truth, it's not a new question. It's a question we've been asking a long time in ecology. How does biodiversity shape ecosystem function? When you frame it in this way, it begs applied questions. We know microbial life represents the majority of species diversity on this planet. Soil inoculation work is really exciting to me because it's an opportunity to use biodiversity as a new class of biotechnology. And by doing so, hopefully better deliver yields, restoration outcomes, and all the other services we ask of our ecosystems. With that, I'll hand it back to you, Martin.
Thank you, Colin, very much for this nice insights how Funga is approaching this topic. And I'm pretty sure there will be plenty of questions, but without further ado, I would like to hand over to Adriana. Please, the floor is yours.
Thank you so much, Martin. And yeah, thank you so much for this invitation to share my work in tropical inoculation. And yes, I'm going to try to describe very quickly what I have been doing. I am currently with the SPUN, but I'm going to be sharing this work that I did as a professor at Universidad del Rosario in Colombia. So my first, I'm going to share two experiments. One was with Koziol uvipera that was carried out by Luisa Fernanda. And I want to tell you a little bit about the rationale why we decided to do these experiments. So in a previous work that we have published last year, we realized we took over 200,000 herbarium records from all the Colombian Andes, and we realized that only 0.6% of the species in Colombia, which is only 6 out of over 20,000 species, are ECM associated. So when we started thinking about this experiment, we didn't know what species we wanted to work with. And this was done in collaboration with Professor Santiago Madriñán, that is the director of the Cartagena Botanical Garden. They were doing seedling production for restoration experiments, no, for restoration practices. And I started looking at the list of species that they were reproducing. And one of them caught my attention because I am an expert in tropical ectomycorrhizal associations, Cocoloba. Cocoloba is a tropical ectomycorrhizal species that often that lives in lowlands. So I asked them, do you know that this species forms ectomycorrhizal associations? And they were like, no, we have no idea. So I realized they were reproducing this species on the same soil that they were reproducing all the other species, that was just commercial soil that more often has non-ectomycorrhizal inoculum because of the low abundance of ectomycorrhizal plant species in Colombia. So we set up to perform an experiment on this species. This is the location of the study site in the north of Colombia. And then we wanted to test two things. One is the germination process. So we took seeds and germinated in peat and sterile peat and native soil. And then we transplanted those to commercial soil and native soil from under Cocoloba trees. And replicated that with the ones that were germinating in native soil. We wanted to know the legacy effect of the first substrate that touches the seedling, that the seedling is germinating on, and how the second transfer where the seedling goes. what we found really quickly is that cocoa inoculum did help seedlings grow faster, bigger, and also the ones that were germinated in native soil and transplanted to native soil, or even if they were transplanted to commercial inoculum, they grew better than the ones that we reproduced in peat initially. We also found that we sequenced the roots. We found that actually the seeds that were germinated in native soil had more colonization of ectomycorrhizae. And also here, we don't have some treatments because they show absolutely no sign of fungal colonization, and they didn't amplify when we run the regular fungal primers on them. So that's like a big take-home message. This inoculum is important, and this specific inoculum is important for this species. Then we set up another experiment in this endangered species, Jugla neotropica. Jugla neotropica is known to be arbuscular mycorrhiza, but in my previous work as a postdoc at UF, I have gone to all over the world where there are Juglandaceae, and I have sequenced the root microbiome of all Juglandaceae genera. So I know a lot about the Juglandaceae inoculum and or root microbiome, and I realized this species is very special because in Juglandaceae there are a clade that forms ectomycorrhizae, and juglans really have very specific fungi associated to its roots, also because it produces juglone that is non-allelopathic, and that could affect also a type of fungi that I can associate. I did this with my students, Mariana, and this is the location of the experiment. And here we have to work with material that was already being produced for the restoration experiment. And we grow it in just normal, regular soil, and that they were using for reproducing everything else. And we also use species-specific inoculum in some of them, only 6%. What we found so far is that, yes, actually inoculation helped plants grow better, but also not all inoculum were equal. We really track what individual juglan, adult juglans, the inoculum came from, and we realized there was one inoculum source that really have an effect. We also found that the origin of the seeds matter. The seeds that were local to this place where we collected the inoculum had higher responses to the inoculum. That's seeds that were from a different population from far away. And I'm gonna leave it there. Thank you so much.
Thank you very much, Adriana. Very interesting data. I would like to hand it over to Liz. Please, Liz, the floor is yours.
Thank you. Seems to be weird on my end. How does it look on your end?
We don't see it in presentation mode.
Okay. Sorry about that. Just a second.
No problem.
Works so well in the test. How does that look?
Yes, you're good to go.
Okay, great. Sorry about that, everyone. So you can call me Liz as the panel goes along. And I'm going to be talking about the good, the bad, and the dead on arrival of inoculants. So I thought I would start by talking about some of the good and some of the work I've done in the tallgrass prairie of the US. And so my work looks at doing restorations of these threatened ecosystems. And what we do is move not only seeds, but microbiome transplants in our restoration. And we found that moving these old growth microbes works great to improve diversity, productivity, and stability in these systems. And so why does this work? Well, in our disturbed soils, we do a lot of things that can really disrupt fungal communities, such as tillage, brownfields. Lots of human impacts can change the abundance and species composition of the fungi in those soils. So if we take a microbial community from this disturbed site and grow native plants in it, we see they do okay. But if we grow those same plants with their whole microbiome soil from the prairie, they grow about twice as well. And we also have found that this is driven by just the arbuscular mycorrhizal fungi from these old growth sites here in this black bar. So we see the same effect with just AM fungi as we do with that whole microbiome transplant. And so that's great because we can culture AM fungi in the lab and use it as a restoration tool. So the next question I wanted to ask with what can I do with these old growth AMF is how much do we need to put out into a restoration to see an effect? And so I'm going to show you some data from a 7th year restoration trial where I had plots where I put out anywhere from 0 to about 8,000 kilograms per hectare, like literal dump truck loads in the plots. And so I'm showing you 2 sets of data. And with both of these data, we came up with this number of about 128 kilograms per hectare is the optimal amount of this stuff to put on to have an effect. And that's because there with this non-native abundance, it declines with fungi, but right around 128, it sort of becomes consistently low. And right around 128, we see lots of late successional plant establishment, and those are our native plants we were trying to restore. But restoration isn't the only application. Crops are also planted in these disturbed soils. So we wanted to ask, can we use these old growth native AMF, which would have been at this site, but now it's disturbed, to help crop growth? And so I'm not going to jump into the data from all the crops, but we've looked at a ton of them. And here are just some pictures where of a few select crops. On the left, they have our old growth fungi, and on the right, they are a sterile control. So you can see these crops are also pretty responsive to native prairie fungi. So we've learned a lot about when and where native AMF can improve plantings. And the key takeaways that I want to impart on you is that we are always using diverse mixtures of native AMF. So sometimes we have as many as 21 different isolates. And in all the studies I presented, those are our native old growth fungi that we isolated from remnant grasslands. And so this is why I started my small business, which is called Microbloom, which provides locally adapted AMF inoculants for use across the US only. We've done one project in Canada at this point, but very local, because I really believe in the power of native inoculants and the benefits they can provide in systems. But the inoculant industry is massive. It's huge and growing, and there's dozens, if not hundreds of different products hitting the market every year. So I want to show you some data from a study where I wanted to ask, how good are these inoculants? So I did a global meta-analysis looking for any paper where scientists tested 5 or more commercially available AMF products. And I was able to include about 250 commercial trials in this meta-analysis where they looked at crop growth of various crops and also root colonization. And so in summary, about 4% of these trials killed all the crops that they were testing these products on, which is a bummer. And across all of these different inoculants, about 84% of them were non-viable, meaning they put it on plants and there was no colonization of AMF in the roots. Furthermore, about 50% of these non-viable products, so dead on arrival products, were beneficial. And this is due to the labeled, and in some cases, some people have found unlabeled fertilizers in these products, which can be really, really high. So the other thing I do, and I wanted to bring awareness of, is I'm a curator of INVAM. And INVAM is a source where you can go to find high quality fungi. So it's the world's largest collection of arbuscular mycorrhizal fungi, which moved to the University of Kansas in 2020. And we maintain about 1,000 isolates of fungi and around 100 species. And we have fungi from all over the world. And INVAM's collection is not-for-profit. It's available to anyone for a small fee, researchers, public people use it for outreach, and commercial products as well. Another thing we do at INVAM is quality assessments, most often for commercial industry, like viability assays. And we've worked with companies of all sizes, from brand new startups to some of the biggest companies in the world. And I've had a lot of disappointing interactions in my role at INVAM with companies. Some companies are great, but many companies really push for weedy, easy to grow strains of low diversity. Some companies have absolutely no knowledge of AMF biology and how to care for them or even assess if they're alive or not. Really, what's really disappointing is that we've informed some companies their products are not viable and they continue to sell these dead on arrival products. So there's this big mismatch between all this awesome science we have showing how inoculants can work to address some of our global challenges with food production and in restoration. And that definitely is in mismatch with the public's experience using these commercial products, which are most likely dead on arrival. And this will continue until we have some regulation on this industry, which currently there's essentially none. So with that, I'll stop sharing and happy to take lots of questions on that.
Thank you, Liz, on the insights of these commercial products and also the, the importance to customize the inoculants to the actual site. So then we will already move to our last panelist, which will be Miranda. I'm looking forward to hear your side on the topic. The floor is yours.
Hi, everyone. Can you see my slide? Yeah.
Perfect. Yes,
Okay.
we can.
Awesome. Yeah. I'm glad Liz segued into what I'm going to talk about, and I'm not going to talk just about regulations, but I'm going to start there because she's right. There's a ton of great research about how and when and where and what good biofertilizers can do, but there's an absolute dearth of research about the risk they can do. So that's what I'm going to talk about it. And I'm not here to, trash anyone's commercial dreams or to say that biofertilizers are all bad. I just want to ask some questions that I think need to be asked. So the regulation of biofertilizers in general, no matter what country you look at, those that have regulations about licensing these products, they say, are they viable? Which Liz showed not really. Are they what they say they are, right? Do they have the right organisms? What else is in there? And the other thing that they usually or sometimes require is evidence that they do promote growth or they do promote nutrient uptake. But these studies are generally done in like very artificial greenhouse conditions. So that's kind of what is out there. What I'm suggesting we need to ask are questions that make sense if we're dealing with live organisms, which we are, and potentially invasive live organisms. So the question should be, do they persist? If they don't persist, if you put them in your field and they die within one season without reproducing, maybe they're not such a big risk. Do they compete with native fungi? I mean, they're live organisms. And probably most importantly, what are they doing to ecosystems outside of, well, maybe inside the crop? Just because they promote plant growth doesn't mean they're not affecting other ecosystem processes on a larger scale. So these are the questions I think we should be asking, and these are the questions that aren't being asked. So I'm going to just give you a snapshot of some of the research we're doing in our group. First question is, do they persist? Now, these are pictures of sites in the Okanagan where I live, and 10 years ago, I started an inoculation study where as a new faculty member, I inoculated these wild ecosystems with a commercial inoculant. Let's just move on from that and I'll tell you what I found. These slides show the number of plants that were positive for the commercial inoculant over time. What I want you to notice is that in the 10th year after inoculation, at one site, over 50% of the plants tested positive for this isolate. And at the other site, it was more like around 30%. It sticks around. But not only does it stick around over time, in natural ecosystems, which are undisturbed, that's the important part about the study, is that the abundance of them. Now, if you look at the abundance, this is the abundance of the commercial fertilizer over time. And it looks, wow, like 2020, there's almost nothing there, right? However, if we kind of do some calculations about what these numbers mean, even at their lowest in 2022 or 2015, actually, that still works out to about 80,000 spores per liter. That's a lot of fungi. I mean, it looks low, but that's enough fungi to have a serious footprint on what's happening below ground. Okay, so yes, they persist, and they persist at least over 10 years. What about, do they compete with native fungi? Now, I want to tell you about one particular study a student of mine is working on. And just to put it in a frame of how I'm approaching these studies, I want to look at them. There's been so much research over the last few years about intra-isolate or intraspecific variation among AM fungi in particular, right? They share like maybe 50% of their genes within a species. This is terrifying to me because one of the biggest rationales for using biocommercial products is, oh, it doesn't matter. That species already exists. It's cosmopolitan. Doesn't matter if we just augment it with a commercial fertilizer or biofertilizer. Well, these isolates are really, really different, so it might matter. Okay, so I'm just setting you up for the study. The study is done by my grad student, Selena Spence, and she grew 2 isolates of Rhizophagus irregulare, and then she sampled individual spores from the pot to see whose spores were they? Who did they belong to? Overwhelmingly, the spores belonged to the commercial isolate, 197, 198. We found no spores belonging to the other isolate, which was not a commercial product. Okay, that's pretty worrisome. However, she found 3 spores that had both genotypes in them, suggesting that when they were grown together, they were able to anastomose, and both genotypes ended up in the spore. Now, this particularly I find worrying because of the common mycelium network. If we add these isolates to environments and they were able to anastomose with the existing common mycelial network that would exist in a natural system, can they then really quickly colonize, spread, invade a system by hijacking the common mycelial network? Who knows? We haven't done this yet. These are just baby steps in this area. But clearly they do compete with native fungi or other fungi. Okay, so what about ecosystem consequences? Well, this is work by another graduate student of mine, Joya Carson-Austin, and she created mesocosms with different plant species in them, and then she inoculated with different isolates of Rhizophagus. And here's just one shot. There's 3 different isolates and a control with no isolate. And I just want to show you that 197, 198, which is the commercial one, did so much better on Bromus inermis, which is a very weedy plant. There are differences. There are differences in how these isolates promote plant growth to the point where some taxa didn't grow at all with some of the isolates. So she's just analyzing these data right now, so we don't have any figures for you. But it looks like there are consequences on plant communities, depending on what isolate you use. Now, whether or not they affect ecosystems, we can't say yet, but stay tuned. We're doing transcriptome analysis to see actually how these soils are functioning differently after an inoculation with a foreign isolate. So, wrapping it all up, what do I want to say? I want to say that soils are perhaps the most complex systems on the planet. And the approach so far has been so reductionist. What taxa can we use? What can we grow? What can we apply? It worries me. It worries me, and it worries me about the stability of these ecosystems trying to manipulate the soil communities by a handful of taxa. I think there's so much more we need to learn before we can start doing that. Thank you very much.
Thank you very much, Miranda. Very exciting topic, and I agree, quite often overlooked. So with this, we have heard the insights of our 4 panelists, and with this, I would like to ask all panelists to turn on their camera. and their microphone. And so we will start with the Q&A round. And please, dear speakers, if you have anything to add, don't hesitate, just jump in anytime. Okay, so I think I had a look a little bit at these questions, and I think what is coming often is, okay, that we can agree if an inoculant should be successful, you know, maybe it has to be customized to the environment. But the question is, how do we select native inoculants? I mean, where to look for those, and how do we select the ones that are successful? Maybe Liz, I think you might start with this, and then other ones can take over. I mean, where to find the successful ones?
Yeah, so I guess our approach with our old growth fungi, so we've done experiments all over the US, but most often in tall grass prairie. And so what we do is we go to an old growth or a remnant site to collect our original inoculant source. So we can go in and take a soil core, you know, just like a 1-inch soil core, and from that core, get a ton of spores of these old growth fungi. And when we're culturing our AMF, we can do that in the lab with other soils to bulk propagate them. And so we don't have to resample this old growth site anymore. So I think it's pretty sustainable. You do have to disturb the soil in these few reference ecosystems, but we can get fungi from them and propagate them in the lab over and over again without returning to disturbance. And our approach in terms of distance, we always try to find a remnant within that state, at least, if not within that county, we can sample. And then we grow super local inoculant for our restoration projects for that. There hasn't been a lot of work looking at how local needs to be. I think there definitely should be some more with that. Another resource people have is the InVam collection. We have isolates from across the globe, 49 countries. So there's a pretty good chance we at least have some isolates for your country if you're thinking about starting an inoculant. I think that's a much better place to start than just picking the random weediest, you know, Rhizophagus irregularis in the collection or something from who knows where. So yeah, both of those things I think should be considered.
Thank you. Any other input on this topic?
I'm happy
Maybe
to share.
Colin?
I think the other thing that can be really important if your goal is to work with native organisms that are in and of the place is actually like having an understanding of like what is the real native biodiversity of the system you're working in. So whether that's tall grass prairies or pine forests, right? Like if the only way we can really know what intact biodiversity of these systems are is by survey, classic biodiversity surveys, which we can do with environmental DNA. And by doing that, you can get a better understanding of what quote unquote should be here. So you can do things that are closer to restoration rather than trying to apply the same organisms everywhere or organisms that are totally exotic.
I have something to say about this. I think it's tricky. I think I totally agree with what Colin and Liz said from a restoration point of view, but the truth is most of the people using these products are growing them in managed systems, right? So this place-based solution for crops that have been bred or genetically modified in the last few decades, they're probably not going to do well with these old growth communities. And that's what farmers won't want them. So we're going to, for this industry to exist, we're going to be forced to create these weedy isolates to use in commercial applications. So there's no easy answer. There's no easy solution for that.
Is that obvious though? I guess what I find really inspiring about Liz's work is her demonstration that actually a lot of these old growth fungi can have effects that are positive for a lot of these commercial plant species that have been bred. Am I right there, Liz?
Yeah, we published some work on peppers and tomatoes, and we have some with hemp and echinacea, some perennial crops as well, that they can benefit from old growth fungi. But their benefit is smaller, for example, than what we see with our threatened prairie plants, you know? And so the benefit to a native ecosystem, I think, is a lot stronger than the benefit you will have when you're growing a bunch of basil, for example, because basil has been bred to have a lot more roots, for example, and exist in these soils that don't have good fungal communities. So I think they can benefit, but that benefit is slightly smaller than in native systems.
Thank you very much. And with this, maybe we can move a bit on to the next logical question. Once you have identified, hopefully, the inoculant that would work, how do we, first of all, bring it into the system, and then how do we actually measure best if it has established in the system and if it's actually doing the job that it should do? Again, that's an open question that came up in several different questions. Whoever wants to go first.
I can start with how do you measure them, because you can't. So, quite simply, you can't measure them because the high throughput metabarcoding analyses we do could, in theory, in the future do that, but none of the target regions we use can differentiate among isolates. So we don't know when we put a Rhizophagus irregulare in and we do metabarcoding, we don't know if we're getting the native ones or the commercial ones. So right now, it's unless you develop individual probes for your isolate, we don't know if it's established, and we can't.
I think some people who've worked with whole community inoculations and restoration systems, Jasper Wubbs and his team come to mind, have compellingly shown with metabarcoding that while they, like you're saying, Miranda, they cannot track isolates, but they can show the new novel community members they've introduced are establishing and persisting, which I think is pretty compelling. And so in that context, if you're talking about introducing organisms in a restoration context that used to inhabit this landscape that don't anymore, and you've actively introduced them, you can track those things with metabarcoding. And do they work for plants? I mean, that's a straightforward measure plants sort of task.
We spend a lot of energy measuring the plant response. So in the prairie, we have all these plants we know are crazy sensitive to their old growth prairie fungi. So we have plots in the field where we add some and plots where we don't. And we see a lot more of these plants establishing when we give them their microbiome that they would have been adapted to. So we're often looking to the plants for this answer because, yes, it is such a challenge to sequence these isolates. I think there's a group in Czechia that started doing packed biosequencing, and they've had some luck with like monitoring individual isolates, but they've only tried it a couple of times and it also fails too. So it's really hard to actually track these microbes once you put them out there. I don't remember the part of the question, but besides
also if they are viable or not, and that you cannot really then get through the DNA tracking, basically. Okay, so but let's hope I mean, the plan is that they can establish, but how to bring it in? I mean, are there specific formulations that need to be considered? Do we bring in spores, mycelia? What do we bring in? Like, how does it work to be successful?
So in the US on inoculants, and I'm talking about AMF inoculants specifically, that you can write propagules on your product package, and that can refer to anything. I think California now has started to regulate what propagule means, and it has to be a spore. But in the past, people have used it to meant hyphae, for example, which isn't something that you can put in a package necessarily, and it is viable once you put it in the field, you know, up to years later from when that package was created. So I think for AMF inoculants, spore has to be the thing we consider a propagule. It's also something we can assess really easily to see if those product claims match what is in the product. There's been some work, including one of my paper, looking at these propagule counts, and they're incorrect up to 100%. And on average, it's about 90% incorrect, and overestimates of the number of spores that they have in their products. So that's a challenge in terms of how much to put out there. A lot of these products, even if they are alive, the number you might be putting out there doesn't necessarily match the label. So that's tricky. And in terms of what we should be thinking of inoculating with, in terms of native restorations or in terms of using different commercial products across the globe, In a lot of the work we've done, we've tested tons of isolates. You know, we have 60 prairie isolates of AM fungi. We have 1,000 in the InVan collection. We've used them in a number of different contexts, including different trials where we have a whole bunch of single inoculations, double, triple, you know, up to 21 different isolates applied in some of these trials. And consistently, the diverse mixture tends to work the best. So that's what I always recommend to people. If they're thinking about an inoculant, is to try to look for something local if you can. That's a huge challenge in the world. But diversity is really important.
I think the same argument I had for the last comment applies to this one too, right? If you're using them in broadacre agriculture versus trying to restore an old field, you're going to need, first of all, not all fungi really germinate that well from spores, so that's a problem. But the truth is, we don't know. We don't know what you're doing to the field, what that does to the inoculum you put in, right? If you put in an inoculum that's totally viable and it totally matches the crop you're trying to grow, but then you fertilize and till, then what are you doing? What are you doing to your inoculum, right? So I think that the applied versus the restoration have probably really, really different recommendations.
Maybe I wanted to say something here
Yes,
to
please,
show
Adriana.
something that, for example, arbuscular mycorrhizal systems might work really differently from ectomycorrhizal systems. That's important to know. So know your system very well. Many of these questions are only focused on agriculture, which totally understand is like a huge sector. But yeah, responding that question of what we should inoculate with, for example, for arbuscular mycorrhiza, we recently know that they rely on bacteria, for example, to produce phosphatase, things like that. So are they also matching the bacteria that help them? And how, I just want to show the importance is not only diversity of mycorrhizal fungi, is diversity of the soil that really makes the effect in sometimes, and that's why it's also important to inoculate to try whole soil inoculum, which is a huge risk as well because it has pathogens, it has a whole ecosystem on it. But in some cases, it could have some advantages. For example, the ectomycorrhizal systems are crazy diverse. And for example, in the tropics, we have found a lot of endemism. It's just very interesting to see the difference between systems, between questions. What Miranda just said, agricultural recommendations are so different from restoration, for example. But I was just thinking how we can mix this together. Could we recommend I don't know, do some native inoculated plants in between the crops to continue to have those native communities, for example? I think just we just had to think more about the ecology of these systems. What is the right way to continue having this diversity? Their complex interactions continue to happen over the time, and of course, exotic species and invasive species are a huge risk in ectomycorrhizal communities. We see it all the time as well. So it's just, I'm really enjoying this panel a lot and learning things.
I'd love to follow up on what Adriana just said. I like, this idea, how do we integrate native biodiversity into these very large managed landscapes? Agriculture represents something like 50% of the arable land surface. It's generally not considered a biodiversity solution, but there's interesting things happening all the time. Huge amounts of work trying to create native pollinator habitat in agricultural landscapes to support those native pollinators, but also deliver ecosystem services. I've heard work about people working on native prairie strips integrated within row crop agriculture that create reservoirs for pollinators, for fungi, for things like this. And I think people like Liz's work, showing that you can integrate native mycorrhizal fungi into agricultural ecosystems is really interesting as well, right? Like if these, these systems are going to be there for, the food and fiber we need, but to what extent at all can they also act as reservoirs rather than deserts of biodiversity? And I think microbes can play a big role there. And that's the approach we take at Funga as well.
I just have one little point I want to throw out there thinking about across biomes. So for that meta-analysis I talked about a little bit, our original goal was to also include other types of inoculants like ectomycorrhizal fungi. But we only found one paper out there that assessed mycorrhizal products 5 or more. And so we couldn't include it. But so yeah, so there's a lot of work that needs to be done in that system for sure. But that one paper did show similar findings where a lot of the products they looked at weren't super viable.
I think that's actually an interesting topic. So in that range between a single inoculum and basically the whole soil inocular, which might be difficult to upscale in the end. So somewhere in between there are these synthetic communities or these assembled consortia. But how to approach this, because they might compete with each other, like how to select, what are ways forward to select these synthetic communities, these consortia, to make them successful? There are many questions in that direction.
I think, is that the question you want us to address, Martin? Yeah?
Yes, please. And I think also the risk is obviously becoming much more difficult to assess them as well.
I mean, I think this is something that we can figure out for horticulture, right? I think we know what the plants are, we know what their genotypes are, we know what the growing conditions are, we can control the growing conditions. We can probably come up with a handful of fungi, at least, if not other organisms, that are really reliable, representative, and promote growth. That's not a problem. The problem is trying to do this in natural systems. And all of the speakers today have talked about how important places and how important co-evolved relationships are and how much complexity there is. I am very pessimistic, and I know Colin and Liz will not like this. I'm very pessimistic that you can have anything that's useful outside of a very local community. I think you can, like what Liz is doing. I think you can do that. You can have these local reservoirs that are going to promote local communities, plant communities, but I don't think we can have this in a generic kind of commercial sense that's going to be easy to capitalize on.
I think you're right, Miranda, that if our hope is to apply the same organism or group of organisms everywhere, we should not expect that to work. And I think that's been shown pretty well. I think it's possible to take a locally specific approach and geographically tailored approach. That's what we focus on at Funga for many reasons, but mostly for biodiversity reasons and also for function reasons, because that's what we think is actually going to work. That's what's supported by the evidence. Doing that well requires a huge amount of, greenhouse and field trials. And I don't think that's been undertaken by the inoculant companies we've seen to date. Generally, there's a handful of greenhouse trials, maybe a couple of local field trials, and then it's like product done, sell it everywhere. And I think we're going to need a very different approach to the research and development and application of this work. But I think that's possible. I think we just haven't really had the chance to do that science yet.
I mean, is that what we want? I mean, we want place-based solution. I think I want that. So I don't know. I don't, no matter how many studies we do and no matter what communities we identify that are going to be robust across systems, I don't think we should go that way. I think we'll be losing something if we do.
I think just to clarify, I agree. We shouldn't be putting the same community everywhere. And I hope I communicated that.
One thing I ask people to stretch their minds and think about this comparison a little bit. So in where I'm at in the grasslands, central grasslands of the US, the tall grass prairie, there's a ton of restoration projects going on for many different reasons, roadside applications or fields that aren't productive anymore, they want to restore. it's a pretty big industry, and a lot of people do restorations in this community. And to meet that need, there are tons of nurseries that grow local ecotype seed for regionally specific restoration products. I think that can be transferred to microbes. Yes, microbes are harder to work with. You need more training and, you know, microscopes to work with them than you do a sunflower, for example. But 40 years ago, there were no local ecotype seed nurseries, and now there's hundreds just for this ecosystem alone. And so getting those partners on board and thinking about, if you are using local ecotype seed, and that is so important, why cannot also local ecotype microbes be considered for these projects? And there has to be a need, and then businesses will develop that, come and meet that need. Of course, Michael Bloom can't do it all. I'm not gonna, you know, do your local ecotype mycorrhizae for whatever project, but, you know, I hope I inspire other people to form similar types of businesses, and also for other biomes.
I totally agree with that. And Colin, I agree with what you said too about that there isn't the drive to have a single consortium widespread. What about the communities that no longer exist? What about the communities that have already been corrupted by cosmopolitan or human disturbance, and these communities no longer exist? I'm thinking of you know, really rare ecosystems. What do we do? What do we do for them?
I mean, short of resurrecting extinct species, I think we're out of luck. Totally. Yeah, I think we got to work with what we have and our best understanding. But I think the other thing we also know so for example, in our system in pine forestry, right? Like, we work with trees that are native to that range and historically inhabited that landscape that are now used in commercial forestry. But through our research, we've shown that, you know, there's this massive decline post clear cut. And because we've sequenced so many forests and found so many forests of varying ages, we could show that really takes somewhere between like 35 and 50 years for these fungal microbial communities to recover. The average rotation length until clear cut of these forests is like 15 to 25 years. So that they're not gonna recover.
How do you know they're recovering? Like, this is fascinating. I'd love to know more, but how do you know they're recovering? Because nobody, up until 10 years ago, we couldn't even tell. We have no baseline data anywhere of any real value, right? From what microbial communities like were before. So that is another huge challenge, is what are we shooting for? What are we aiming for? And what do these systems really need?
Yeah, I mean, we have to take our best approach. And this is a big question in all of restoration ecology, far beyond microbes, right? And, you know, one approach is always reference ecosystems, right? To the extent that there are intact habitats remaining, we can get estimates of what plant community composition historically was. And this is some way we create restoration targets. We can also look at, you know, space for time substitutions and look at biodiversity accumulation over time, both alpha and beta, and try and estimate gamma. And try and understand it that way, because at least you can understand, are there species that seem to be walking towards extinction without an active approach?
I wanted to add something here, to just add another perspective, and is, for example, and more associated with the consortium question. For example, in the tropics or in many places that are undersampled, we have a lot of species that we haven't even described or don't have names, don't have isolates. So I just wanted to bring up that in many, many places, we're working on a lot of missing data. So it's important to fill those gaps and understand, yeah, what is the effect of bringing those exotic species, and what are the best isolates for that specific region, and really work on the basic ecological questions that are gonna be the building blocks to get to a more useful and safe way of using mycorrhizae for management. And I just wanted to bring something up that is just really concerning, because there are people out there doing this, selling these products. And yes, I just want to support that that will to regulate those things. But yeah, also to improve. I really admire the work that you guys do, because it's just so important to move this forward, making safer, but also try to just create products that people can use instead of using those other ones, because I don't think we have a way to stop these companies, right? So how do we offer an alternative that is at least a little safer?
Okay, so maybe if you allow, I would like to quickly come back to this how to bring it out, because I mean I see many questions are very specific. You know, how many spores to apply, what kind of formulation, how to apply it actually in the field. Do you do multiple times? So please, participants there, email the panelists directly if you're having these very specific questions. But I'm wondering more like coming back to how it establishes. Can we identify key components in a soil system that are very crucial to understand for the establishment, like physical or chemical properties? And I think one interesting or multiple question also go in the direction, what is the role of the soil fauna in actually establishment of the inocula, in dispersal of the inocula maybe too? Has anyone looked at this? And what are these key components to see that it actually establishes?
Who wants to take that one? I have nothing to say, because I think we know almost nothing about fauna and how important they are, and that is critical. But anybody, anyone?
I
agree
think
a little
about
bit, Fauna. Go ahead, Liz.
I was going to say, I mostly just think about A and F, but I do know things like worms can help move these things around and disperse them, right? But worms only wiggle so far and so fast. In terms of dispersal and spread, I know Miranda has led a lot of this work, but there hasn't been a ton. We've done some work trying to track our isolates, and sometimes we can see them, sometimes we can't. Our sequencing methods are pretty crude for this group of fungi. There's been some work saying they might be able to move about a meter a year, which is pretty slow.
But if we think about, you know, more managed systems, and now we go, you know, away from the native, we go maybe to some arable systems, maybe permanent one, like vineyards and orchards and things like this. So they're already quite a bit far away from these native systems. I mean, don't we need to understand what are the key factors in the soil or in the system to, in the end, also understand why or why not these native inocula can establish?
Yes. And I didn't present any of these data, but I work a lot in viticultural systems, and same products have completely unpredictable behaviors. And whether or not that has to do with product quality, like Liz was talking about, or it has to do with the physiochemical properties of the soil, which 100%, it has to do with all of the above, which is why it's so frustrating working in this area, because the consumer base wants recommendations and wants to know, oh, I have a soil pH of this, what should I use? And it's like, well, your soil pH is this, but what are all these other factors? And can we do this giant meta-analysis and figure out what you need? No. I feel kind of fatalistic about this, because I don't know that we'll ever be able to come up with strict recommendations other than, here's a system that has persisted for millennia. Here are the fungi and the plants that persisted together. Let's go use those together, which is what Colin and Liz are talking about doing.
I think there are a couple places where we know, active inoculation programs can be especially effective, and they tend to be the most degraded places, unsurprisingly, right? Like if lots of things are missing, there's something meaningful to restoring something, and so like ex-mineland landscapes, I'm not surprised that Liz sees, very, very big outcomes in the system she's shown pictures of, right? Like where the soil has washed away. It's one of the reasons we chose to work in forestry, because soils are very degraded given the way they manage it, at least in the southern US. Beyond that, though, the soil physiochemical stuff, climate stuff, all the things you're mentioning, Martin, I mean, I think doing that and answering that question well to date has been a challenge of replication and statistical power. I mean, to answer those questions, you need to replicate these trials in space in different geographic locations that vary across all these conditions a huge amount of times, like dozens or hundreds. And that really has not been done. But if it was done, I think we should expect to see some
I don't think
signatures.
so, because I'm replicating these experiments across a very regional scale. Like, and we can't controlling for, we still get such hyper variable responses even within a really kind of homogeneous growing region. So I don't know that it's a matter of scale. I think it's a matter of, like I said at the beginning, the soil environment is incredibly complex and it's incredibly heterogeneous at a small spatial and temporal scale. So unless we can get our brains and our data power behind kind of understanding the level of heterogeneity in soil ecosystems, yeah, then it becomes a statistical power.
Yeah, I mean, no doubt that the heterogeneity is real and very important. I think, you know, in our European work, just looking for correlational signals, though, you know, we had a very big signature of the fungal microbiome and productivity rates. We did perform power analyses. We found we couldn't reliably detect that very large signal until we had sequenced something like 60 or 80 for us. And so, and then when you get to manipulative studies, it's very possible you need to go even bigger. And so when you're talking about that much variability in climate and all these other things, many of which are co-varying, you do need huge amounts of replication to get there, which is challenging, but also to me kind of exciting because we generally haven't achieved that, which means there might be a lot of possibility out there.
And considering, you know, these inoculants or different consortia that people might put out there, remember, some of these products have huge amounts of labeled and unlabeled fertilizers, up to 40 times background NNP. You know, just that alone can have huge effects on what your grapes are going to do over, you know, 5 years, for example. So I always, especially for researchers, if they're going to be using a product, I now recommend they do a greenhouse trial in sterile soil, just plus and minus, to see if that product was ever even alive. And then you can even start to ask the questions, is this because of the inoculants or just a bunch of, you know, very expensive fertilizer, phosphorus that I'm putting down in these trials? So maybe not super relevant to all the participants here, but for the scientists using inoculant products, definitely do that.
Very interesting. Yes, if we actually already talk about, you know, fertilizer or maybe synthetic fertilizer, if we move to even more managed systems like really cropping systems, I mean, is there a negative correlation with actually the level of fertilization and the ability of inoculant to establish? Does anyone know this? These were also a couple questions. I mean, do we, as we move towards cropping systems, from native to cropping systems, do we actually lose that ability that inoculant can establish?
I don't know the answer to your question, but it did make me think of work coming out of Marcel van der Huygen's group, which has shown that, you know, when they work with a very specific strain of AMF, they can predict its inoculation performance based on the background microbial community at that site. And, you know, plant pathogens are a big predictor. And so the higher the load of plant pathogens at the site, the more likely that inoculant is to perform. But I don't know the answer about the fertilizers.
I think the literature, just the historical literature is really concrete about the role of soil nutrient levels and establishment success. I'll turn off my mic.
Yeah, in some of the trials we've done in pots in the greenhouse, you really do have to put tons and tons of fertilizer to see that reduction in colonization, like far beyond what anyone would ever put in the field. You know, there's always fungi in the field. there'll be different fungi than, you know, in your native system, or if you didn't disturb it every year, but there's going to be fungi there and they'll be able to colonize.
Okay, and maybe a very provocative question. I mean, okay, when we talk about inocula, we talk about biofertilizers. Is there a potential that we are actually more harmful with doing this than, you know, in the past with applying agrochemicals? And can we stop it even? I mean, is the regulations in place that there's not a Wild West in how this is going to look like in the future? Are we moving in the right direction or not?
I don't know we're moving in any direction. I sit on a lot of panels talking about regulation, and I don't see us moving anywhere closer to regulating. I mean, yes, we can get it. We are in a Wild West situation. We can move out of it by having really robust policy, which is possible. However, like I said before, they're out there, and they're reproducing in natural ecosystems. So maybe this isn't the end of the world, but it is something to think about.
I think when we think about a restoration context, it's also really important to consider the counterfactual. Like, if we're losing these ecosystems, we're almost certainly losing microbes, and what happens if we don't do it, right? And I'm not saying it's obviously one direction or the other, but I am saying that there's cost on both sides. And we need to consider that. Yeah, that's what I'll say. Liz.
So just thinking about that meta-analysis, it was pretty much the same in the US and Europe, Australia, Africa, the continents that I found data from. The pattern was the same. Between 80 to 90% of these inoculants were dead. And that gives me hope that we haven't caused as much damage as we could have at this point, because these products are, you know, so terrible. But in theory, these businesses will get better at making mycorrhizal products. I guess we haven't seen that in the 20 years that meta-analysis encompassed, but in theory they will. And yeah, that it is really concerning. And it's extremely concerning that there are no regulations. You can import mycorrhizae spores from anywhere in the world to US, and lots of countries have similar things. And producers have no obligation other than self-reporting on their product quality and consistency. Something scary in one of the papers we presented, we found that 5 out of 16 US inoculants had a bunch of pathogens in them, you know, so we need regulation. I myself have emailed a few senators, both in the state and nationally, and have had 0 responses on my concerns. So as a scientist, I don't know where to go to help facilitate regulation of these things, but yeah, we really need it.
Why do you think that is the case, that we have these regulations in place for many agrochemicals, you know, and they are getting, you know, I mean, more and more strong on this, but on the other hand, here it seems like it's not moving forward. Is it just that the big pressure of the companies, of the big companies, to see the new market with biofertilizers, biostimulants?
No, I don't think so. I think the only time you really see robust policy is when there's human toxicity involved. And I think these aren't ever going to kill a person. So perhaps that's why no one cares.
But there are some regulations on like seed movement, which is a propagule. So I'm not sure why this propagule is not the same as that. I think it's just harder to work with. So they don't, you know.
Yeah, I mean, absolutely. Whenever there's a pathogen that's going to destroy a crop, right? That's why we have all of these phytosan policies. But these are mutualist, right? They're supposed to be mutualist. They're supposed to be beneficial. So I think it's just the whole.
Sorry, it's also lack of knowledge about fungi and fungi ecology, right? We know we regulate seeds because we have been working with plants for a long time. We know that they could become invasive, and it's maybe a matter of educating the policymakers about how fungi work and that the potential risks. Maybe they are not really thinking about this and they don't understand. And I think that's an important part of our job, to try to educate people. Yeah, I was gonna say something else, but I forgot if I remember.
I think that's a good I think that's a good point, Adriana. I think the burden of proof is always post hoc, right? Did it do any harm? I don't know. Let's figure it out. We still, you know, maybe I'm wrong, and we will find out shortly that there is no risk to ecosystems. Maybe there's replacement attacks up, but you know what? At ecosystem scale, it all kind of balances out and everything's fine. We don't know yet. We just don't know if there are any consequences. That's what if there are consequences and we can go to legislators and say, look what happened to native pollinator networks or whatever that the legislators care about. I don't think it's pollinator networks. We need to show them evidence, but we haven't done the research yet to even show them that.
My
I
approach.
think that-
Oh, go ahead. Okay. My approach, right, was to not really talk about the fungi, but to talk about the money. So we have estimates of there's global AMF inoculant industry is like more than $1 billion this year. If that data from the meta-analysis is extrapolated, that's like $800 million this year people are spending on nothing in terms of mycorrhizae. So I try to communicate that, but it also still seems like it hasn't been working. So if anybody has any ideas or knows any regulators who might care, I would love to talk to them.
But at which level?
Oh, go ahead.
Sorry. No, go ahead, Colin.
No, it hasn't stopped them with supplements either, which is an even bigger industry. But I mean, I think all of this speaks to the importance of like, we need to try stuff. We need to understand what works, what doesn't, where, why, right? Like, does soil inoculation work? I think the answer is resoundingly yes, sometimes, but we don't know when, where, why, and all the consequences. And I think it's on us to find ways to do that research. And I think that's becoming really interesting in the current political environment. And I mean, one of the reasons we chose to start the company was because we weren't going to achieve the levels of replication and scale we thought we really needed to understand these things. And so this is our one vehicle to do this. I think there's probably many more. Love SPUN. And I think that's how we're going to build the evidence we really need to be compelling on this front.
But at which level do you think regulation should take place? I mean, is this that each country would do it? Should this be orchestrated by the FAO, or how? Where should that take place? That level of regulation?
Well, it depends on what level of government is able to restrict the flow of these products, right? So I think most of the agricultural policy is at the federal level in Canada. I don't know about the rest of the world.
And maybe something else that is also coming up. I mean, in the end, you know, we humans, we like to think in boxes, and then we have the good ones, we have the bad ones, the pathogens, right? But in reality, biology works a bit differently. So there's a continuum, and they can obviously switch lifestyles. They can go from something that you think they are good to become a pathogen, depending on the condition. That seems to me very difficult to deal with. It's very unpredictable. I mean, how do we approach the biology that is there and that, yeah.
I could say something here. One thing I wanted to say from the previous conversation is that I think and is related with your question now is one of the reasons why these haven't been implemented, probably in some we haven't having like this big shocking results is because of the complexity of the system. It's a very diverse system, and the responses at the species level, for example, are very context dependent, and also the plant mycorrhizal or plant fungi interactions are also very specific and very context dependent. So that, yeah, complicates things a lot. So I believe that local research should be done in order to create local policy. I think that would be ideal because we have seen this is not one size fits all, and we really need local information to be able to inform local decisions. And something else that I wanted to say from before between harm and good is that based on what I have found in my tiny experiments with native species, is that in some specific cases, when you have very good ecological knowledge of the system, the inoculation is necessary for the plant to survive and to protect the local soil biodiversity. So in some cases, yes, we are doing more good than harm. But of course, in the one size fits all, you are doing more harm than good. And we are in a gradient too of good and bad.
I think that what you just said, that last sentence, that could be the soundbite for this whole conversation, right? One size fits all is more harm than good. Yeah, I love that.
Oh, Martin, you're muted, man.
Yeah, now I'm back. Thank you, Colin. Okay, so we're already slowly approaching the end, but I have one more question to put it a bit into bigger context. So it's a very promising area. It's also a controversial area of research where we need to do a lot of thinking, how to do it, how to regulate it, how to keep the risks under control, how to avoid the overselling, I guess. But in the bigger picture, I mean, how much is soil inoculation really one of the best solution, for example, for restoration compared to, should we just focus on better practices in the end? I mean, where can we put that into perspective?
I think this is a question that's asked a lot in restoration ecology more broadly. Like, at the end of the day, there's this spectrum between passive and active restoration. Some places will naturally regenerate and dispersal processes are functioning. And when that's the case, that is absolutely the solution. It's far cheaper. You're going to create an ecosystem that better resembles intact forests and natural regeneration processes. It's awesome. Some places do not regenerate and absolutely need an active restoration approach if those ecosystems are going to recover on any reasonable time frame. And I think that's very likely true in microbes as well, especially when we're talking about extremely disturbed environments that are frequently disturbed. And we know this from mine reclamation studies. And so I think the thing we're all driving towards is actually really this predictive understanding of where is this active and passive restoration approaches, like where should we be leaning on which and when. We have this for plant community ecology to some extent. I don't think we're there yet in below ground ecology, but there are some obvious places where active approaches can be very important.
And maybe I wanted to add something else, and is that there has been, in restoration, there has been a huge bias to above ground diversity, and that focus on underground diversity has been really missing. And for example, people bring plant material with soils from outside. They are actually inoculating microbes there. They are not thinking about it. So we really need to take an underground focus on this restoration. And I totally agree with what Colin said. Some ecosystems really will benefit from reintroducing the microbes as well as the plants. So we just need to think about plants like host species for microbes. And we don't, or restoration practitioners, due to this bias, have not been thinking about this. And I think it's really important to start thinking and being more conscious about what we bring in this restoration for underground purposes.
So that maybe then would be the last one. If it is promising, and it is, right? How do we make sure people pick it up, like the practitioners, the foresters, the farmers? I mean, are they willing to just follow the evidence you lay out, or is there skepticism towards these approaches? Maybe if everyone could give their insights on how this is perceived by the practitioners, then we can finish the event.
Where we work in forestry in the United States, there are key influencers that hold a lot of sway here, and they happen to be the tree nurseries who supply the seedlings. They happen to be their consulting foresters, which are often, attending actual meetings to keep their certifications up to date, and their research cooperatives at universities. And all those are places and opportunities to influence these communities of practitioners. And so I think, while it's difficult to shop forest owner to forest owner, right? Like, there are these high leverage points, and fortunately, those points are very open and interested in evidence, and in my experience, like, are open to very new things. They're excited about it if you come to them and acknowledge, with humbleness, that this is their field, not yours.
I agree. There's incredible amount of enthusiasm in my discipline. The growers want to use it. They're so enthusiastic. They want to use all of it. But the problem, as Colin mentioned, is the influencers are largely representatives from the companies. So they're getting a very one-sided message. So we need to get this information. I know the Americans are great in that they have this extensive outreach system at universities. We don't have anything like that. So we need to get the information in their hands so that the growers are getting a balanced story.
Please,
And maybe
Adriana, the final word.
Yeah, just a final word is that, for example, from my experience in Colombia with small restoration, they don't even know about the importance of mycorrhizae, that there are several types of mycorrhizae. So we really need to start from scratch. I think education is the key. And yeah, the education should be done by the experts that are going to show both sides, dangers and the good things. And we just need to give the whole story out there.
Please.
In terms of communicating, yes, there's lots of outreach that can be done. I think as scientists, we can publish our work in forms that are open access, so practitioners can read it who don't have subscriptions to Wiley or whatever very expensive place that our work might end up. To access can be a barrier for some people to even start to learn about microbes. So think about that as scientists. Some practitioners have had really bad experiences because they have used some of these DOA products. And so that can be a barrier, and has been a barrier for some of the people I've worked with, and trying to educate them on what is different about our approach in our lab with native and old growth microbiomes, and how that's different from a commercial product requires a lot of conversations. I've had numerous different talks where I've given, some of this data on how awesome AMF are. And then the farmer is so excited, and he goes out and buys the cheapest mycorrhizal product he can find and puts it on the field, and then is super disappointed with the results. And yeah, that can be a challenge, talking about all the different diversity of products and microbes and outcomes that can happen.
I think that's an excellent end point here, because if we talk about communication, outreach, and education, this is really what GSBI is all about, right? We want to inform people, bring it to everyone how important soil biodiversity is. So I think that's a few excellent last statements. So with this, I would like to close that event. I would like to thank a lot to the panelists. That was very, very exciting to have your different views on this and your input presentations. I also would like to thank the audience, the participants. Many have joined. There were many, many questions, way more that we could answer. So we could be here, I think, for 24 hours and still not have answered all the questions. So thanks for placing these questions. And with this, I close. I wish everyone either a good day ahead or a good night, depending on where you are on the globe. And see you next time.
Thank you, Martin.
Thank you so much.
Bye-bye.
Thank you. Bye.
Bye.